Welcome to the second instalment of microbial metabolism in Bio 1104. There's references there, so you will see by now that we use a lot of different images, so the images will have their web address with them as well. So in this one, we're going to continue looking at the biochemistry of microbes, some of the metabolism, and that's because we utilise some of those outcomes for identification. And you'll be able to get quite a good idea of that when we do the prac sessions, okay? We're going to be looking at some of the basic molecules and look at enzymes, how they catalyse reactions, and then go into some phosphorylation and light and dark reactions, okay? So we're going to compare and contrast aerobic and anaerobic respiration. We're going to look at fermentation. So everyone's probably had yogurt in their life, or cheese, or wine or beer. So you'll see what the end product of fermentation is like. We're going to look at lipid and protein catabolism, so the breakdown of those, and just use some tests, identification tests, to show how they're utilised. Look at some of the other pathways and compare and contrast. So it's basically looking at the different pathways and comparing them. So why do we care? Well, you really do need a very basic understanding of biochemistry to appreciate the results that you get with identification tests for microorganisms. And you also get an appreciation of how certain bacteria can cause a disease. So we'll look at fermentation. So you think fermentation, that's a good thing. Some types of fermentation can cause gangrene. Okay, so you have to be aware that there are quite a wide variety of outcomes. Then with the identification, I've got some really quite good pictures of triple sugar iron, one of the agars that we can use to actually identify what the fermentation is and if there's any gas production or H2S production. Okay, so we'll have a look at that. We're going to look a little bit at fermentation, so dairy products, cheese basically, and how microbes can survive in particular environments. And you do need an idea of biochemistry to look at the nature of immune responses. So if you think biochemistry is those molecules that can be changed, that happens in our body. Okay, so enzymes are particularly important and binding is particularly important with your immune response. So if we look at the structure of biological molecules, and some people who've done chemistry, this will be quite familiar with them, but it's basically living matter has carbon, okay, and carbon is the main thing, but you also have hydrogen, oxygen, nitrogen, phosphorus and sulfur, and there's trace amounts of other things as well. And then for them to have an action, they have what's known as a functional group that's attached to the structure. And those functional groups can be carboxylic acid. So some molecules you might see written out longhand with COOH at the end, that's a carboxylic acid. Okay, so acetic acid is one of those. You also get lipids and amino acids having that same functional group. Then we've got amino functional groups, and these are the NH2 groups. And typically they're amino groups, so they're found in amino acids. And everyone knows that amino acids are building blocks of proteins. Then there's the hydroxyl groups, the OH groups. These are the ones that you find in alcohols and glycerol. And sulfhydryl groups, the SH. And you have in proteins, proteins have a very globular structure, so they're not just one straight chain. They're globular, and you will be able to see a bit later, they have different indentations where enzymes can fit in, or vice versa, okay? And for them to keep that globular structure, then they need bridges. And some of those bridges are in the form of sulfhydryl bonds. Then we've got organic phosphates and ester linkages as well. So that's what you see in the ester linkages in triglycerides. And we'll be talking about triglycerides. Sometimes there's three, and there are three fatty acids attached to a glycerol backbone. Okay, sometimes they're called triacylglycerol, okay, or tags. So there's just a few names for them. So if we look at the enzymes, so enzymes are proteins and they catalyze a reaction. So if you look at the diagram down the bottom, we've got this purple bit, which is the enzyme. And it's a particular enzyme, and it's named basically for what it does. So if these two substrates were ADP and phosphate, this would be known as ATP synthase. Okay, so these two substrates bind to particular sites on this enzyme, and when they're brought into close proximity, then the enzyme substrate complex actually allows that reaction to be catalyzed. So it actually decreases, having an enzyme in there decreases the activation energy, so the process or the chemical reaction can proceed really quickly. Okay, so instead of waiting around for a long time for them to bind together, etc., come together, they're facilitated by this enzyme that catalyzes it. And you can see here this representation. You've got the purple and blue bit, which is the enzyme, and here we've got these substrates just going into that little pocket in the enzyme. Okay, so it brings them together, gives them a surface to react with each other. And that is really helpful because you need enzymes to move reactions along in the body. So things like photosynthesis, you have really nice formula. So carbon dioxide and water plus light energy gives you glucose and oxygen. So that's quite easy to understand. But when you're producing energy, you actually need some particular processes catalyzed as well. So when we have this type of reaction, we get the oxidation of glucose. So remember, we're moving electrons around to produce, basically the energy is trapped in ATP. And ATP is our source of energy in the body. Then with glycolysis and the production of pyruvate, then that can actually happen both in an aerobic and an anaerobic environment. So if you have aerobic, it will be, as we expect, plus the production of energy. But if we didn't have oxygen, then this would still be catalyzed, but instead of going to CO2 and water, it would move into lactic acid. Okay, so without oxygen, it moves to lactic acid and energy, or it might go to alcohol, ethyl alcohol and energy. So you can see the fermentation there? Yes? Okay, so just to be aware that same starting product, same starting substrate, but depending on the environment, it can move in different ways. Now that's important to understand because when we look at microbial growth, they can either grow in oxygen or without oxygen, and some in between because they're microorganisms and they do that. So if we start, we've looked at aerobic respiration. So if we start with anaerobic respiration, this is the one that uses electron acceptors other than oxygen because oxygen, excuse me, is not in the mix, okay? And they actually produce less ATP or less energy than the aerobic variety. So things that might be used as electron acceptors might be nitrate. So it's reduced to nitrite and then it can go right down to nitric acid or even to nitrogen gas. And this is what happens with some bacteria, so Pseudomonas and Bacillus. So sometimes if you have an anaerobic incubation and you open it up, sometimes you can really smell that nitrogen gas. So you can actually suppose that it could be Bacillus or Pseudomonas. Sulfate can be reduced to hydrogen sulfide by some bacteria, such as Desulfo vibrio. Now, other bacteria produce H2S as well, and I'll show you what that looks like a bit later. Carbonate can be reduced to methane, and everyone has smelt methane? Yes? So sometimes, if you walk into a micro lab, you can smell a variety of smells. So this is just a quite nice little diagram showing you that there's the electron and what is the product, what the outcome is. Okay. Now fermentation, you probably all love cheese, so this should be of interest to you. Fermentation is the release of energy by fermentation of sugars or metabolism of sugars, and it only involves glycolysis. Okay, so if you remember the Krebs cycle, etc., the electron transport chain from last week, that doesn't happen in fermentation. So what happens? You have glucose as this, the substrate. You get the production of pyruvic acid, but instead of going through the Krebs cycle and electron transport chain, it goes into lactic acid that we've seen earlier, or ethanol. Okay, so it can go in a couple of different ways. Now, please remember that oxygen is not required for fermentation. And yes, and there's those two ATP being produced there. Now, the two important fermentation pathways are the homolactic fermentation. So this is when pyruvic acid is reduced to lactic acid, and that's what happens with lactobacilli. So they're the bacteria that produce our yogurt. Okay? Some of the lactobacilli, and depending on the species, they can be used for producing different types of cheeses as well. Okay, but it's basically pyruvic acid goes to lactic acid. Okay, and you have the lactobacilli doing that. Alcoholic fermentation is when you remove CO2 from pyruvic acid to form acetaldehyde, and it's anaerobic. Okay. Now, acetaldehyde is reduced to ethyl alcohol, and that's what is produced by the action of yeast, such as Saccharomyces, to produce wine. Or some Saccharomyces are also used because they produce the CO2. They're perfect for providing that rising in bread dough. Okay, so you also have a little bit of ethyl alcohol being produced as well, but that's when you bake the bread, then that is removed. That's probably the better way of saying it. Okay, so this is great. So we've got the lactic acid, the homolactic fermentation producing the cheese. We've got the alcoholic fermentation producing ethyl alcohol and CO2. But we've got other fermentations occurring as well. Now this one is the butyric fermentation. This is the one that causes gangrene. Okay, so if you have the main causative agent of gangrene is Clostridium perfringens, and when it enters the tissue and there's no oxygen, then it can undergo that particular type of fermentation and cause that necrosis of the tissue. Okay, so it's kind of juxtapositioned with the lactic acid and the wine. Sorry about that. So we've looked at carbohydrate catabolism. So we've looked at respiration. So we've got the Krebs cycle and the electron transport chain. And compared with that, we've got the fermentation. And so you can see that it doesn't go into the Krebs cycle and doesn't have an electron transport chain. So these are basically the comparison between the two. And just to remind you about what the electron transport chain does, you get that production of ATP after a number of electron transfers. Everyone okay with that? Yep, okay. So other processes that you can have are things like metabolism of lipids or proteins, and we're also going to be looking at photosynthesis. And so if we look at lipid catabolism first, so catabolism is breakdown. Yes? So the enzymes that help with that breakdown or the hydrolysis of the lipids are the lipases. Lipases break down lipids. And these are the triglycerides. So if you look up at this top image here, we've got a glycerol backbone and then 3 fatty acid tails. Now they can be the same fatty acid or they can be different fatty acids. Okay? So, but what happens is the lipases actually hydrolyze the lipids and break off that glycerol, and then you have those 3 free fatty acids. Then this glycerol goes to glycolysis, and the fatty acids, they are there to react with coenzyme A, and they're basically, with repeated oxidations, they will go into the Krebs cycle, okay? So the smaller bits go into the Krebs cycle. So you have that energy. Now, these are really good storage molecules of energy. And when you start to break down fats, you get a lot of energy being produced. Now with proteins are hydrolyzed, they're broken down to their component amino acids. Now Everyone knows what amino acids are. So they're the building blocks. And you know we've got about 20 or 21 in eukaryotes. And 9 of those are essential, which means we have to get them from our diet. The rest our body can make, but those 9 are essential. And so the amino acids for protein synthesis, they can be recycled. So if you break down protein, you get the amino acids, the component amino acids. They can be recycled and reused to make new proteins. Or they can undergo hydrolysis as well. So you get deamination, which means you remove that amino group. So that NH2, that's why it's deamination. You're getting rid of the amino group. And those molecules can be used, what's broken down ones, can be used to synthesize new amino acids, okay, or they can go into the Krebs cycle. So there's a number of different pathways where they can go to. And so a summary of catabolism. So if we have food, it may consist of fats, carbohydrates and proteins. We have seen that the glucose and other sugars go through glycolysis. The fats are broken down, fatty acids and glycerol. They're beta oxidized and go into the Krebs cycle. Proteins broken down to amino acids, and they can go into different parts of that glycolysis and Krebs cycle as well. But the outcome is energy. Okay, so you get different amounts of energy from each of those, where with fats being the producers of a lot of energy. So that's the breakdown pathways. Now the anabolic pathways, these are the ones that are building up. And so if you're building up something from lots of components, it requires energy. And that's exactly what happens here. So you need the energy and the metabolites to combine to form a product. And that's when you are putting together carbohydrates. Maybe you've got a glucose molecule and you've got lots of excess glucose molecules and you want to store them. You put those glucose molecules together in a form like glycogen that can then be stored in the liver or skeletal muscle. Okay, so you are synthesizing that carbohydrate. You can also do the same with lipids. So if you've got an excess of fatty acids, for example, you can start metabolizing those, putting the fatty acids together on a glycerol backbone and storing them as fat. Same with protein, you can build that up from those amino acids. You also need nucleotides. So everyone knows we've got DNA and RNA in our cells. So how are they synthesized? And that's what is known as the nucleotide biosynthesis. So if we start with the biosynthesis of carbohydrates, the main one is we can have the production of new glucose, so gluconeogenesis, and that generates glucose from non-carbohydrate substances, okay? And it's basically the reverse. And these synthesized molecules can be formed into glycogen in bacteria, glycogen in animals, and then sometime, if it goes down to the fructose 6-phosphate, then they can be used for peptidoglycan in bacteria, which is found where? Do you remember where you find peptidoglycan? In the cell walls. Yes? Okay, so these are very, very important for the storage of glucose, but also for the integrity of the cell wall in bacteria. Now with lipid biosynthesis, you're building up that lipid, and it's so you need both glycerol and fatty acids, and you can see where they come off that that pathway, and then when they join, you get those simple lipids. And as I said, these fatty acids are not all the same. They can be the same three in a fatty acid, and that gives the name of the fatty acid, or they might be two similar ones and one different one. So there's a lot of different structures that you can have with fatty acids and indeed fats. And so you can see where they come off that that pathway. Now with the protein biosynthesis, we're dealing with the same thing. We can get the building blocks, those amino acids, to make protein. And obviously if you don't have those amino acids in your diet, those essential ones, then some proteins cannot be made. Okay, and that can lead to disease states. So you can get them from a couple of different pathways, and with amination, so adding an amino group or taking away an amino group or changing it around, then you can get the production of amino acids. And so with the transamination, you just basically change things around, change where that NH2 is. Okay, so if you start with glutamic acid and you add some another substrate, you can get a change with that amino acid being put onto another molecule. And therefore, that makes a different amino acid. Okay, so transamination is a very important thing because obviously your body doesn't want a glut of a particular type of amino acid. It wants to have the facility to change the types of amino acid, because you're always going to be changing, especially in the human body. And in the human body, you need to be building up different types of protein at different stages. But we all need, every cell needs some nucleotides. So those nucleotides are the building blocks of RNA and DNA. And you get the sugars from the different pathways. So you can get from the pentose phosphate pathway or the Etna-Dordorf pathway. You can get these glycine, glutamine, making the purine nucleotides, or you can go through the pentose pathway, so it can make either pyrimidine or purine. So the two different types that we have are purine and pyrimidine. So you don't have to worry too much about that, but it's just to realize that they are different structures to join together and then produce that coding in your DNA. Actually, probably this one. So we've got the sugar molecule, a phosphate moiety, and the bases. And if that base is adenine or guanine, these are known as the purines. And if it's cytosine or thymine, spelt wrongly, are the pyrimidines. Okay, so that's the definition of them. So if we look at the biosynthetic pathways, which is what you need to have a handle on, you've got fatty acids, glycerol and other components, obviously going to form lipids. And those lipids can be joined with other glucose-like or sugar moieties to form glycolipids. These lipids, some lipids might join with proteins to form lipoproteins. So you've all heard these terms, okay? Same with this, amino acids, they're joined together to form different types of proteins. They can join with lipids or glucose or form nuclear proteins. And why they're important is they're on the cell membrane. Okay? They're the identifiers of the cells, and they can help catalyze different reactions as well. So if we don't just have all anabolism or all catabolism, there's always a mixture of them. So you need to be able to integrate both of those systems. And you can do that through some common intermediates. Okay, so you can see here where those are happening. So some come in, some come out. And if the pathways can diverge, so if it could go one way or the other, they're known as amphibolic pathways. Okay, it means that they can go either way. Now, to just break down, okay, so we've looked at both the catabolism and anabolism, so you know where they can diverge in here. So why do we care? Because we can use those characteristics for diagnostics in microbiology. In the food industry, obviously fermentation is a big thing. Scientific research, we can manipulate different bacteria to produce different things. Sometimes you can get them to produce, say, insulin. So some genetic engineering of bacteria. You can get them, certain bacteria, depending on what they are, they can actually decompose sewage. Okay, so you can utilize what you know about their metabolism and use it for good. But if we're looking at diagnostic microbiology, then we've got two main areas. So we've got selective and differential. So selective growth just means that in the agar, and everyone's familiar with agar now, that solid gel, we can put different things in the agar to select for certain bacteria. So if we put bile salts in the agar, bacteria that are used to bile salts will grow, and ones that aren't won't grow. So bile salts are used in a lot of the Enterobacteriaceae growth. So things like E. coli can be grown with bile salt. Okay, so we can select out for particular populations. Salt is an interesting one because if you think about salt on our skin, we have quite a bit of salt on our skin, sometimes more than others. Staphylococcus species, so Epidermidis and aureus, can tolerate concentrations of salt. So possibly 5%. Staph epidermidis can cope with about 5%, possibly a little bit less. But Staph aureus can grow in agar that has up to 10% salt. It knocks basically everything else out, but Staphylococcus aureus can grow. So we've selected out, because we know what it can tolerate, we've selected that out to grow it. Okay, so we have that selective growth. Sometimes we might want to look at different characteristics of their metabolism. So if you have one type of organism that will produce acid from a particular sugar and one type that doesn't, we can put that sugar in an agar, and if we've got a mixed culture, then we can actually see which will ferment the sugar or not. And we do that by adding an indicator. So if it's fermenting the sugar or metabolizing the sugar, it will produce acid. And if there's an indicator in there, then it will, it will go a different, change the indicator colour, so it will go typically yellow. Okay, so you can actually differentiate the growth. So you can see here that one might ferment one sugar and one doesn't. Sometimes we can differentiate. So this one, I don't know if you can see it very clearly, but this is blood agar. So it actually has agar with blood in it, and I'm fairly sure maybe it's this week, that you will subculture onto blood agar. But you've got some organisms will send out enzymes that break down blood, break down the blood cells. And you can see with this particular one, it's a B there, but there's a clear zone around it. And that's a type of organism that causes beta hemolysis, complete hemolysis of the blood cells around it. Then we've got one that's got the alpha and it kind of looks a bit green. It does. It's known as alpha hemolysis. So it's incomplete breakdown of the blood. And then we've got ones that just grow, but they don't actually interfere with the blood cells themselves. Here we've got, remember I was talking about the triple TSI agar? So here we've got, so typically the TSI agar looks kind of red. Okay, but it's got 3 different sugars in, so that it's known as the triple sugar iron agar. So we've got 3 different sugars and it's got iron in there as well. So we don't have any sustained change in pH here. Here we've got, it's really black. So that means that the bacteria has produced hydrogen sulfide and it's reacted from the iron. Here we've got, it's an alkaline slope and a yellow butt. So we've got acid being produced here. Here we've got complete, so acid is produced in the slant and in the butt, but it also has produced gas. So you can tell a lot from the triple sugar iron agar. Now if we add those together, we've got both selective and differential agars. So if we have, probably we'll go with this one. We've got E. coli growing and producing a particular pigment, whereas Salmonella produces a different colour colony, okay, because it's fermented the sugar in there. So once you get used to the different types of agar, you'll be able to work out what's actually happening in the, if it's selective, differential, or both. Some of the biochemical tests are quite good to use as well. The ones that we're going to be using are catalase and oxidase. So catalase is the one that is present in aerobic bacteria because oxygen can be toxic. So these particular bacteria convert that the toxic oxygen to hydrogen peroxide, but you don't want hydrogen peroxide hanging around either. So then you get conversion into H2O and oxygen. Okay? And what that looks like is if you put a bacteria in hydrogen peroxide and it bubbles, that just means that enzyme is there, so it's catalase positive. It's a really simple test, but you can tell whether it's, by that you can tell if it's aerobic and producing that catalase. So Staphylococcus is catalase positive and Streptococcus is catalase negative. So even though they're both aerobic, one, the Streptococcus, doesn't produce that particular enzyme. Okay, so it's just an indicator. It's another way of sorting out which species it is. Then we've got oxidase. Now it is an enzyme. This particular enzyme, it's cytochrome C. It functions during the electron transport chain and oxidizes the reagent to form a purple colour. Okay, so it's just, you've got special paper that you can put the bacteria on, the colony on, and depending on the colour, you can tell whether it's positive or negative for oxidase. Now the fermentation test, this is a more, a better picture of the TSI, and it, you know how we went through what they are, but with the triple sugar, if you have, so with a slant, this is basically, you've put the agar in a tube and then before it's set, you just put it on its side. Okay, so you get that slant occurring. So you've got a slant, you've got a slant and a butt. Okay, so if you look at yellow, that means it ferments, so that's this one, ferments glucose, lactose and sucrose. Red and yellow, glucose fermenter, black butt. Here we've got the hydrogen sulfide production, and the red is a non-fermenter of any of those sugars. Okay, so you can tell which sugar it's actually fermenting. Now the trick is you can't look at it after 12 hours. You have to do this, you have to read that particular slant between 18 and 24 hours after inoculation, okay, because there is some reversion and that you need that about 24 hours to have the proper analysis of it. Another way of testing if something is fermenting, so you can have a liquid, okay, so these are test tubes. In this bottom image, you've got test tubes full of broth, and the broth has got an indicator in it. So that's easy. So if something grows, you'll be able to tell whether it's producing acid or not. Yes? And therefore fermenting that sugar. But what about gas? If it was producing gas, we'd have no way of capturing it just with a broth. So what you can do is put a tiny little tube upside down in there, and that's known as a Durham tube. And here, sorry, in that first one where there's the red liquid, that Durham tube is filled with that liquid and there's no gas being produced. And you wouldn't expect that because there's been no fermentation, no change in the indicator. In the next one, you've got a change in the indicator, so you've got positive carbohydrate fermentation, but there's no gas in that Durham tube. In the third one, you've got fermentation, so the sugar has been fermented, but you've also got production of gas, and that gas is captured in the up-ended test tube, the Durham tube. So it's a really easy test to do to see if a carbohydrate's been metabolized. Okay, back to plants. Well, not just plants, sorry, I shouldn't be flippant about that. So photosynthesis is the conversion of light energy into chemical energy, and that chemical energy can be used for fixating carbon, okay? And you have two pathways, or two parts basically. You've got a light-dependent path, and then that can be cyclic or non-cyclic, and then you've got a light-independent or a dark reaction. Okay, so what does that actually mean? If we look at the equation, you've got carbon dioxide and water given that light energy, So you've got that chemical, that energy, gives you glucose, oxygen and water. Okay, so that's what we're talking about. But what about how we get that? So if we look just at the light reactions, you've got chlorophyll, and chlorophyll A, alpha A, is used by green plants, algae and cyanobacteria. And remember those thylakoid membranes that we saw last week or the week before? That's where that is found. So in the chloroplasts. And the electrons from the chlorophyll pass through the electron transport chain. So you've got the light coming in here, and then you've got the electron transport chain, and then you've got the hydrogen ion movement. Okay, and so that's what's happening in that. Each of those discs that are, those flattened discs that are in the chloroplasts. And you should know that there's a photosystem 2 and a photosystem 1. Okay, we'll come back to that. And then we've got what's known as cyclic photophosphorylation. So this is when the light energy is captured and converted to ATP without the production of oxygen. So it's anoxygenic, okay? Hence the name. And so what happens is you have the electrons being excited and then going through the electron transport chain, and they come down that excitation level, and then they come down to ground state, back they stay in the chlorophyll so they can be excited again and go through that. So it's a cyclic photophosphorylation. So the same electrons are going around and around. Yes? Hence cyclic. And this happens in some of the green and purple sulfur and non-sulfur bacteria, okay, because they've got that ability. Then the non-cyclic bit. So this is the oxygenic photosynthesis. This is where you get the production of oxygen. Okay, so this happens in plants, green algae and cyanobacteria, and you've got the two photosystems. Okay, so you've got photosystem 2 and photosystem 1. So we're just talking about what happens. You have water coming in, but the water is split into 2 hydrogen and half an O2. Okay, so you get the production of oxygen, and these electrons go into the photosystem 1, go up to a higher energy, go through the electron transport chain. You've got electron transport chain, go into photosystem 1, then go into another primary electron acceptor, and then they are lost, basically. So it's non-cyclic. You can't recycle these ones back here. You have that splitting of water that actually drives the situation, drives the process. And so you get oxygen being produced and the breakdown of water for that. Everyone see that? Yep. Okay. So the types of energy capturing metabolism, we've got the photoautotrophs, the chemoautotrophs, photoheterotrophs and chemoheterotrophs. And so down here you notice that some are in purple, and these are the ones that undergo cyclic photophosphorylation. And the one in blue participates in the non-cyclic photophosphorylation. Okay, so you get the production of oxygen. Yeah? So the next one is the light-independent reaction. And so CO2 is fixed, and when that's fixed, it's used to synthesize sugars. Lord. Okay, and there's 3 cycles. So you have the fixation of carbon dioxide, and that's incorporated into another molecule, and you get, so it goes around in a cycle basically, and it uses energy to do that, okay? And these organic compounds, the compounds that are produced, things like amino acids, lipids, and carbohydrates, okay? Also CHO are carbohydrates. And things like glucose and fructose, glucose, and there's lots of glucose joined together. It could be starch or cellulose being produced depending on where it's being fixed. And then, so the relationship between the light-dependent and the light-independent reactions. So you can see here on the left, you've got the light-dependent reactions. They need light to produce the organic energy molecules. Okay, so you can see those. The light-independent reactions, these are the dark reactions. They actually use energy, and so they fix the carbon dioxide and they produce carbohydrates. Okay, so it's just another compare and contrast. And this is just a summary of the whole thing. So we've got the energy sources. You've got sun or glucose, for example. You've got the carriers of the electrons, and then the final electron acceptors. Okay, so some practice exam questions. Beta oxidation is responsible for, what do you think? Okay, have you got it in your mind anyway? Yep. Okay, bacterial conversion of nitrate to nitrogen gas is an example of? Okay, let's see how you went. Yes? Okay, very good. So, in summary, we've looked at the different types of pathways, including fermentation, looked at some of the applications in identification of microorganisms as well. So you've got quite a reasonable grounding in biochemistry and the anabolism and catabolism, okay, and what enzymes do. You'll be looking, you've looked at cyclic or non-cyclic photophosphorylation and the light reaction, dark reaction, that comparison. Okay, so we might leave it there, and we'll come back